Now in this portion, we'll start talking about special senses. The first sense to talk about is the sense of vision. Now the eye, or the organ of vision, is made out of two different components. There's an optical component, and that is the part of the eye that focuses the rays onto the receptor cells, our rods and cones. And then there's a neural component where the rods and cones are going to transmit that light into a graded and then into a full action potential. We cannot see all of the electromagnetic spectrum. So there are parts of the electromagnetic spectrum that we have no ability to pick up. So we cannot sense radio waves, microwaves, infrared rays, but the part that is visible to us would be this portion, and that is where we can see the visible light. So it's at a certain frequency on the electromagnetic spectrum. But ultraviolet rays, X-rays, and gamma rays are not things that we can pick up. We have no receptors for them. We have no experiences of them whatsoever. Now, a little bit on the anatomy of the human eye. So I'll go through this really quick. More details about this can be found in the textbook. The eye is made out of 3 concentric circles. The one on the outside, or the external portion, is made out of the cornea and the sclera. The cornea is the part on the outside that is transparent, and that is the very first layer that any light ray would have to pass through. It makes the about anterior fifth of the outer layer, and that while the rest is made out of the sclera that is the white of the eye, it's made out of dense connective tissue in order to protect the inner structures. The middle layer is known as the choroid, and that you can see here kind of, that dark orange layer on the inside or in the middle. That choroid has a lot of melanin pigments in order to make the inside of the eye dark, pitch black, so that we are able to see pictures and colors in that perfect darkness of the inside of the eye. So when you look at the pupil, that dark blackness that you're seeing is actually how dark it is on the inside. You are actually looking at the inside of your eye. The middle layer also has the ciliary bodies. These ciliary bodies attach to the lens through these little ligaments. They're known as the suspensory ligaments. And when the ciliary muscles contract, that will lead to the change in the refractory period of the lens. So we could focus different things or change pretty much at this. We'll talk about this more in detail when we get there, but we're pretty much changing the force or the power of the lens using the contraction and the relaxation of the ciliary muscles. Now the third, or the inner layer, that is the retina. That is the beginning of the neural component where we have the receptors. The retina is a layer that remains in its position through gelatinous substance known as the vitreous humor. And so that is the vitreous humor, again, is very gelatinous. It keeps the retina plastered in its position. So if anybody gets an eye injury where the vitreous humor is drained for some reason, the retina detaches very easily. So retinal detachment is kind of an easy thing to occur. Now, all 3 of these layers are going to be perforated towards the back or the posterior part of the eye by this optic disc. The optic disc has the optic nerve and the ophthalmic arteries and the ophthalmic veins. Okay, so the blood vessels. If you look at the retina, okay, through the pupil, you look at the retina, you are going to find these 2 areas. This right here is the optic disc that we talked about. So this portion where you can see the optic nerve and the ophthalmic blood vessels, this area does not have any receptors, so no rods and no cones, and it is also known as the blind spot, so that if anything any light falls on that area, there are no receptors to activate, and hence the name blind spot. Now, we also have this area that's a little bit darker than the rest of the retina. That's known as the macula lutea. And all in the very middle, there's your fovea centralis. Now, the fovea centralis only has cones. Cones are the receptors for perfect colored vision, very sharp pictures, and so on. As you go further away from the fovea, you'll start getting more rods until you reach the peripheries of the retina, and those are full of rods only without any cones. Rods are responsible for dark vision, so you use those when you are out in the dark. They pretty much see grays, shades of gray. The images are not very sharp, so you can't really tell shapes and colors and so on. So this is where you want the rays, the light rays, when you're focusing on something. This is where the image would lie, or would the light rays would focus onto this fovea area, the macula in general. Anything that you could see on the peripheries of your field of vision would be lying on the peripheries of the retina. Okay. Now, what about the focusing point? How do we focus light? First of all, a little bit back to physics, that when these rays change or go from one medium to another, so say from air to water, for example, or they have to pass through glass. If they are, where did this cursor go? There we go. So if the rays are at a 90-degree angle, no refraction will happen. So you see this ray goes through, nothing changed. But if the rays are at an angle with the surface, these angles are going to bend. That bending is known as refraction. Okay, and that is what happens in our eyes. So the cornea is the first surface to refract light, followed by the lens. We cannot adjust the power of the cornea, though, but we can adjust the power of the lens. And here again, you could see that we are focusing these rays on the fovea area, and that will give us the sharpest, most colorful image, while everything else that will fall on the rest of the retina will give us, the blurriness that we see on the peripheral vision. Another thing to take note of is that what we see upright will go onto the retina upside down. Now, when we take a look at how vision reaches the eye, there is a way to flip this back so that we see everything upright and we're not seeing everything upside down. And when we get there, I will mention this again. Now, remember we said that the cornea is not adjustable, so we cannot adjust the refractory power of the cornea, but we can adjust the refractory power of the lens. So under normal circumstances, when you're looking at something from very a far distance, these lights tend to be a little bit, more or less parallel to each other. Not much has to happen, so the cornea and then the lens will refract those lenses or those rays, and they'll go and focus on your fovea. Okay, so here the ciliary muscles are relaxed and the lens has not changed. But when you bring something very close, those rays are now diverging away from each other, and you see here that the rays are out of focus. To bring them back and focus them back onto the fovea, you would have to increase the refractory power of the lens. And you do that by contracting the ciliary muscles, increasing, pretty much squeezes the lens, and increasing its refractory power that is mediated through the parasympathetic nervous system. And you can see here that the rays are now focused. So I'm going to go ahead and let's watch this little video. There's no voice to it. It is just showing you how we adjust the lens. So what we're looking at here, this is the lens. These are the ciliary muscles, those red processes on the side, and these right here are your suspensory ligaments. So you can see here how the ciliary muscle is going to contract in order to increase the refractory power. There's the contraction. We increase the power. Now they relax, and that decreases the refractory power. Okay, so now back to our slides. And where did they go? There we go. Now, how do we also correct vision defects? What are vision defects? So under normal circumstances, all of these rays are actually focusing on the fovea. But in the case of nearsightedness, also known as myopia, nearsighted patients are able to see near things, so they have no problem reading, but they cannot see distant objects. Okay, so you can see here, this is normal sight. In nearsighted individuals, the rays are focused before the fovea, so by the time they reach the fovea, they are all messed up again. And you will correct that using a concave lens. Now, in the case of farsightedness or hyperopia, that's a little bit different. That's the exact opposite, where they can see from afar, okay, but they are unable to see near objects. It's hard for them to read. And for those patients, you would prescribe convex lenses. So you see here, this again is your normal sight, the rays converging onto the fovea. In a farsighted patient, these rays, but they need more time. So at the fovea, they are all messed up again, and they give them these very blurred vision. You would correct that using a convex lens. So I have a little bit here on what is astigmatism and a couple of questions on what is cataract. So astigmatism would be blurred vision due to the bumpiness of the cornea. So it doesn't have anything to do with the lens. It's all about the cornea not being smooth. That would lead to astigmatism. Cataract, on the other hand, is the cloudiness of the lens. So normally the lens is made out of different proteins, and those proteins are transparent. As we age, so there are diseases that cause it, but most common cause is age. As we age, these proteins get older. They tend to start to get calcified, and it's hard for the lens to change its refractory power. And then eventually it becomes totally opacified. You could see it kind of looking like these clouds in the eye. Okay, and it is now considered part of the normal process of aging, where the lenses are removed and replaced with new ones. Now let's take a look at the organization of the retina. So the retina is made out of different layers, and we have our rods and cones, and then we have bipolar cells. Remember, the bipolar cells were the cells that were neurons of special senses, where you have the cell body with tools coming out of it, hence the name bipolar. And then we have ganglion cells. There are other cells, but these are the ones that we're going to be focusing on. As the light goes through the eye, the light actually goes in this direction. So it hits the ganglia first, and then the bipolar, and then the rods and cones first. These, though, do not have any pigment in them, so they have no ability to pick up light. The rods and cones do. So these are the receptors. Again, okay, so your rods and cones are your receptors. These are the rods. These are the cones. And they have a pigment. We'll talk about the pigments in another slide. They'll pick those up. These are they will lead to a graded potential. Because it's in a receptor, we call it a receptor potential. And that potential will move on to these bipolar cells. Then the bipolar cell will take that graded potential and go to the ganglion cell. Now, the ganglion cell is the first of all of that, these three cells that we talked about that have the voltage-gated ion channels. And because they have voltage-gated ion channels, they are able to produce a full action potential. So these are the first cells in the pathway of vision that have the voltage-gated ion channels and are able to perform an action potential. These, the rods and cones and the bipolars, they lack voltage-gated ion channels, and they can only go through graded potentials. Now a little bit more about the photoreceptors. So remember we said we have the rods and the cones. Cones are for color vision. Rods are for black and white or night vision. Each part is made out of an inner segment and an outer segment. These outer segments have these little shelves. So you can see these shelves. These shelves are known as discs. And you can find photopigments in them. So these photopigments are made out of vitamin A. It's a vitamin A derivative known as retinol. And it's different from rods to cones. So in rods, the photopigment is called rhodopsin. And these are very sensitive to light, while the pigment in cones are known as opsin or conopsins, and those are sensitive to red, green, and blue light waves. So not just those specific color, but it's more of a range of red. Okay, so the different grades of red, different grades of green, and different grades of blue. And that's why we're calling them these different light waves, okay, because it's not just one color of red, it's different shades of red, and so on. Now, what is the neural pathway of vision? We already said that vision is going to come from the side of the ganglion and then bipolar, and then hits the rods and cones. But again, it's the photoreceptors, those rods and cones, that have the ability to pick up vision or to pick up the light. The photopigments change their chemical composition and are going to lead to a graded potential. Remember, these rods and cones do not have voltage-gated ion channels, but that graded potential is going to transform over to the bipolar cells. Same with the bipolar, they do not have voltage-gated channels, but it will transfer that graded potential to your ganglion cells. Now, ganglion cells, again, are the first cells in the pathway where an action potential can be initiated because they do have these voltage-gated channels. Photoreceptors, there's a whole bunch of interaction between the photoreceptors and the bipolar and the ganglion cells. There's a lot of on pathways and off pathways that we are not going to get into in detail unless you are preparing for a PhD of the photoreceptors. I will be happy to go over this with you, but until that point, we just want to know that some receptors are going to be turned off, others are going to be turned on, and this is how we see the different colors and the different 3D images and so on. Now the neurons are going to go through the visual pathway until they hit the visual cortex, and that is in the occipital cortex. What we did find also in the ganglion cells, we found a third kind of photopigment, and that is known as melanopsin. That is going to send signals to the suprachiasmatic nucleus found in the hypothalamus. So if you remember that term, that nucleus was responsible for the biological clock and our biological rhythm. And that is why when you want to fall asleep, you want to, calm down, you want to turn off the lights. You do not want to be looking at your cell phone. All of that light hitting the eyes will stimulate the ganglion cells and to break down melanopsin, and that is going to stimulate the suprachiasmatic nucleus, tricking yourself into thinking that it's still light out there and you still need to be awake. Okay, so obviously the that nucleus again and mediated through that pigment helps in the keeping up of our daily cycle. We are messing it up though, looking at our cell phones throughout the night, but that is how normally it would have been. Okay. Some visual information would have to go to the brainstem and the cerebellum. So although visual info goes to the occipital cortex, but there are parts of the information that needs to reach the cerebellum and the brainstem. Because remember we said that we integrate different information. Part of the information that we would integrate, for example, when we're walking, would be where are the things in relationship to me so I don't start bumping into chairs and so on. So you want the cerebellum and you want the brainstem to know what you're looking at and where they are in relationship to you. Now this is the visual pathway. So we already talked about the retina being activated, the optic nerve. We also have the these are the optic nerves, and they cross over at the optic chiasm. So there's some crossover of these fibers to the opposite side. Not all of the fibers cross over, just the nasal fibers. So these fibers that are on the nasal or on the medial side of the optic nerve are going to cross over to the opposite side. And then they have to go to the thalamus. So remember that all of our senses, with one exception, would relay in the thalamus. The nucleus in the thalamus where it relays is known as the lateral geniculate nucleus, or the LGN. And then from the thalamus, it goes through this optic radiation to the visual cortex and the occipital lobe. Remember when we talked about seeing things upside down, it's this optic radiation that flips the picture back upright. Now, color vision is due to the fact that we have cones and these opsins. We are able to detect those three wavelengths that we talked about, the red, the green, and the blue. And it's usually a mix of different things. So we have the ability to differentiate between all of these different shades of different colors and so on. And it's because we are turning some of them on while turning others off in different degrees. And that will give you these different colors that we're able to perceive. Okay, if we are perceiving, or if we're getting a mixture of all of the wavelengths, then we will see an object as white in color. If we are not getting any wavelengths from an object, we will see it as black in color. So black is the absence of these wavelengths, while white is the presence of all of the wavelengths. And as we said, so although each type of cone is excited most effectively by one color, but it's really a wavelength of color, so different ranges of that one color. And we are able to discriminate color. Does not only depend on which receptors we activate, but it also depends on the intensity of light. So if you look at something in dim light and compare that same picture while looking at it in strong light, the colors are going to definitely be different. Okay, and that is because, again, in that stronger light or stronger illumination, you are definitely increasing the amount of cones that are being stimulated. While in dim light, it's hard to stimulate cones in dim light. They're not as sensitive, so you're mostly stimulating the rods, and that would give you a different picture. So in terms of which one is more sensitive to light, rods are definitely more sensitive because they have the ability to pick up the dimmest of lights, while cones, you would have to stimulate them in more bright light. So they are less sensitive to light. Now, what is color blindness? Color blindness is more common in males. It is due to a recessive mutation in the genes that code for these cone pigments. The most common kind of color blindness is red-green color blindness. And it's where they either lack the red or they lack the green cone, or it's a mixture of both. And because of that, it's hard to discriminate between these two colors. It's hard to discriminate between the shades of these colors. And this is one of the examples of what is used in order to test for color vision. Because again, green and red is the most common. This is the one that I'm using. You can see different shades of green, different shades of red. You would ask the patient to ask them what is that number. So if they cannot see that it's a 57, then that might indicate that they have an issue with their color vision. Now for eye movement, we have some muscles of the eye that are inside of the eye. Those are known as intraocular muscles. Those are responsible for constriction and dilation of the pupil, or the ciliary muscles that we talked about that constrict or contract and relax in order to change the power of the lens. Those are intraocular. The ones here are known as the extraocular muscles, and there are 6 skeletal muscles, which means that we can control those eye movements. And those are attached to the eyeball, usually at the sclera, and are responsible for movement of the eye in the different directions. We have the movements basically breaking down into fast and slow. So slow movement would be, you moving your eye looking from one thing to another. Fast movements are known as saccades. And these are very small, jerky movements. You might not even notice them. So if you're watching TV, for example, you are always stimulating the same cones over and over and over again. And again, remember, that can lead to adaptation. So what the eye does, it does these very small, jerky movements to make sure that that every time you're looking at these images, you're stimulating different receptors. So you don't want to fixate the image on certain receptors, leading to adaptation. These saccades also happen during sleep. So there is a stage of sleep known as the REM, or the rapid eye movement, and the eyes are moving back and forth very fast. It is not related to watching your dreams. Okay, it has other functions, but again, this happens during sleep. Glaucoma, which is another pathology that can happen to the eye, and that is the increase in the intraocular pressure. So there's an increase of the pressure inside of the eye that can lead to irreversible damage to the retina, because you're damaging the rods and the cones and the neural components. That would either be due to the increase in the production or the decrease in drainage of the aqueous humor. So I didn't mention what the aqueous humor was, so if you're not familiar with it, let me go back here. So that chamber here between the lens and the cornea is full of a watery fluid known as the aqueous humor. Now, if we make too much of it, or there's something blocking its drainage, that would lead to the increase of pressure of the eye, and that is known as glaucoma. If you've ever been to the eye doctor and they kind of give you that puff of air, that's how they measure intraocular pressure. The causes of glaucoma are not really known yet. It does tend to happen in families, so we do know that there's a genetic component. It tends to happen more in diabetic patients and happens as a side effect of certain medications. Macular degeneration is another pathology, which basically means there is detachment and degeneration at the macula. Remember, the macula is where that has the fovea, and that's where you'll get your perfect image. So macular degeneration is going to lead to that loss of visual acuity. In most individuals, it's related to age, and hence it's known as AMD, or age-related macular degeneration. So this is the eye of Horus, which is not the eye we were talking about, but I hope you enjoyed the eye, and I will see you in our next installment of Chapter 7.